There's this fungus called the Chytrid fungus, and it is a frog killer. So they built this contraption, and it heats them up. And they can survive it. The fungus does not.
SciAnts. We get experimental. SciAnts. We're curious, non-judgmental.
About these frogs that had better not be sad.
So you actually gave a deep dive on this years ago.
This is a Chytrid virus?
It is. I was hoping you could give us that little bit of the backstory intro.
We were just mucking around on the Arizona upper rim. To be fair, I didn't study it. I did collections. I did a lot of conservation. When I was a student, I was like a field hand. I helped with research a lot. Yeah, we were just looking, so we would just like catch frogs and swab them. And then the thing that was a bummer is we'd just go into like this little tiny pond. It was almost seasonal. Not quite, but you know, it's Arizona, so it was almost seasonal ponds. And so they were, a lot of them, and they were just small. And we were tracking this fungus. And so every time we'd go into one of these little ponds, catch a frog, or hopefully a couple, and take some swabs, and then you'd go out, and everything, your waders, everything had to be bleached down, disinfected, and then you could go to the next one.
Because of the likelihood of taking
You just didn't want to risk. We were trying to track the movement of this fungus, and we obviously didn't want to be the cause of spreading it. I mean, we knew it was spreading. Also, again, to reiterate, this was not my research project. I was an assistant on this, just helping. I mean, it was cool. It was really cool work. It was very cool work, but stopping and like doing an hour of deep cleaning, disinfecting in between every single collection gets really arduous. Y'all, you will get tired of it. So, I mean, it has to be done, and that's for science. Like, you're gonna do what you're gonna do, but it's not as fun as like misnetting bats and stuff, let me tell you guys.
So that's what I was asking, so like of the difficulties of doing something like this and why you were doing it, and it's just because Chytrid is so deadly to the frogs.
It's devastating. Yeah, the fungus is worldwide. So this is true. And I mean, even at that time, like this was many years ago. Even then, we knew it was kind of all over the place. We just didn't understand it really. If you read literature for that time period, people are like, we think it's this thing, Chytrid, and people didn't quite understand it, but we knew it was moving through this one area. And so by doing really frequent testing, you could see how it was moving through an ecosystem on this small scale. So it was helpful not just for that ecosystem, but actually, which of course we cared about because like native fish and frogs in Arizona, but actually getting to understand the dynamics of how it moves through an ecosystem like that is a little bit unusual. So it was a good opportunity to be able to study that as well. Okay, anyway, sorry. Yep.
Well, so then the treatments are a big thing too. Like, so a lot of the issues in the Chytrid fungus treatment had been that there's a way to treat the amphibians, but as soon as they let them back out, they just got reinfected. And it's deadly for these frogs, and not just frogs, but other amphibians as well. And so there had actually been a couple of different treatments that they had tried. So I've got a couple of videos, y'all, to go through of kind of what this is, what are some of the treatments that they've tried, and why this new treatment is extremely promising because it's cheap, so it's cost effective. There is an immunity element to it, like an immune memory component to this treatment that isn't seen in other things.
Interesting.
And it's using their natural biology to fight the Chytrid.
Interesting.
It's, I think, really, really cool. One thing that gets buried in this paper is the immune memory component of it. Like, they spend a lot of time just talking about, like, the process of what they built. But
where was this published?
Nature.
Well done, Nature.
High-end publication in Nature. Here's
also, Wombat, fungal infections can be a huge problem in humans as well. It again depends on the fungus. But like looking at you, Kevin, the aspergillus, right? Kevin is actually a fungus.
Kevin is a beautiful fungus.
He is a beautiful fungus, but in immune-compromised people, he can be a highly problematic and pathogenic fungus. But in healthy individuals, he's just a happy little guy hanging out in your compost pile, so no need to worry.
He's also there's a strain of him in soy sauce. But yeah, it's published in Nature, like the big Nature.
All right. Tell me about this paper, please.
We can't get to it yet. We have to talk about all the stuff that doesn't work to make this why it's so cool. All right, let's hear about why Chytrid is bad.
Yeah.
There's a couple of treatment options that have been tried in the past, including vaccinations against Chytrid. We're going to talk about why that doesn't unfortunately work. And I know it's a lot of build-up to this paper, but I think that's also why this paper is in Nature. Right. Because so many other things haven't worked. So it's not y'all just to the golden frog, and it's actually not just to frogs. I think it's broader amphibians, right?
I think that might be true, but it is particularly well known because it is decimated frog populations, potentially causing the extinction of several.
Yeah. There was some in the paper that said like 90% of the population has been taken out, for example, of certain frog species that they were looking at. So I guess that's what kind of you would do, is that you'd go into the water source and look for...
You'd catch frogs and we'd actually swab them.
Okay, so you weren't just looking for the size, like the exoskin,
we
but you have a swab on it.
Yeah.
And this is, y'all, something that we've talked about in the past, of if you, like with the black-footed ferret, if you have an opportunity to save a species... Do you just bring them all inside and start a breeding program? That's what they did with the ferret, right? So then do you bring them inside and save the species and try to cure them, or?
The problem is, listen, conservation biologists never want to do this. They're never like, you know what's a great idea? Let's round up all of the remaining species. We're not great at keeping stuff alive. All right, if they're having trouble out there in the wild, we don't want them in here. All right, I mean, sometimes you have to do it. But a lot of animals do not do well in captivity. We are not clever enough yet to be able to make this always work. So it is really the absolutely last thing that you do. You pretty much play this card if you are certain that no intervention is going to lead to the extinction of the species. Because at that point, it can't get worse. At least maybe you can... get some specimens, send it to the frozen zoo, and you have a chance at keeping the species alive. A chance. But that's why you don't do it. Conservation biologists will not do it unless there's literally no other choice. It is not a move that anybody wants to make.
There's this fungus called the Chytrid fungus, and it is a frog killer. This is kind of the backstory on what it does, like how the skin of the frog starts to expand as it's trying to get rid of the fungus, and the animal eventually can't breathe. And this attempt to save them was to bring them all in and treat them, but if you treat them of the fungus and re-release them, they just get infected again. And so the paper that we're going to be talking about is probably the best treatment that they've identified so far, because it's really cheap. It's efficient, and it confers an immune memory to those that have been infected, which is unlike other treatments.
And when you say immune memory, can you rephrase that so we're all on the same page?
Kind of like how if we get vaccinated, we have antibodies ready to go to fight the infection. They didn't talk about what the mechanism is in these animals, like what is primed in terms of like what's ready in the immune response, but their immune response is essentially ready against the fungus to be able to fight it better, faster than if they've never been infected before.
Essentially this treatment cures them of the fungus and prevents them from getting sick in the future.
Yes.
Okay.
Unlike this treatment here, where they're getting rid of the fungus, but if they were to reinfect, it'd be as devastating as quickly.
It does, yeah. So it's not preventative. Every time the frog it would be exposed to the fungus, they need a whole new treatment. And the one you're going to tell us about, one treatment, and then they, even moving forward, they have a protection against it.
And it's really inexpensive to build. Like, so it's a structure that they build to actually do the treatment, which is really neat. And it's like, they gave in the paper, like, oh, you just get these PVC pipes and heat sources and you're Gucci.
This is crazy.
So like you said, even though they've got all these frogs and they're curing them, they can't release them until there's a plan.
All right, so tell me what you have, what this paper found.
No, we can't get there yet. We have to get to all the background stuff that doesn't work, Lita. sorry, I got very excited by this paper, and I felt like it would be important to look at.
I gotcha.
She's so over. She's just give me the punchline.
Okay, so why did this not work? Was it just too labor intensive?
So that's the argument, is that it's very labor intensive. The dosage is not easy to note.
Okay. So like species to species.
Exactly.
You have to do a whole new set of experiments.
And you have to bring them into the lab. Yeah. They have to survive the lab. Like, they have to infect them. They then have to treat them. They then have to re-release them. And that's a whole hassle and a half. And so it was, if it was maybe just one species, that might be enough to solve, but it's not enough to just do on one. They wanted something broader. This will not induce immunity to the germline. So they didn't look and see if there was any next generational effect.
Like a immune priming or anything.
Yeah, like if for some reason the offspring were just like Lita said, like they're primed and ready to go, or like even a little bit of a more robust immune response. They didn't take a look at that, so we don't know if there was anything, whether RNA-wise, passed on to the next generation to prime them or not. So, like, they were saying, the frogs that were re-released, it's like, yeah, maybe they'll be ready to go, but are they gonna be? Like, what else could be happening? What else needs to be done? And if it's just that population, will the offspring be, there's a lot of issues here.
Is that really effective? One
My
dose
argument
treatment?
would be,
has that been effective? That is not a trivial undertaking. And is one treatment really enough?
Science News of the Week, we're just on a paper that has now this breakthrough treatment of chytrid fungus, which is a deadly fungus that actually wipes out amphibians. Their treatment, Lita.
Yeah, tell me.
So it turns out the fungus cannot survive in high temperatures.
Sure.
And so they built this contraption.
Okay.
Here it is. Here's what it looks like. They have it also with like a cheaper one with PVC pipes.
Okay.
But it's these little openings that the frogs go into.
Yeah.
And it heats them up.
Okay.
And it heats them up to, I think, 6 degrees above like the normal ambient temperature.
Okay.
And they can survive it.
Okay.
The fungus does not. It clears the infection.
It clears the infection.
It clears the infection, and the frogs are immune primed.
Okay, so it's similar to the fungicide treatment where they were robust the first video we watched.
Yes, but cheaper.
Right, no, I get it. I'm saying it's the same principle. They had the infection, they cleared the infection with help from people, and then that protects them moving forward.
Yes.
I love this! This is crazy.
I thought it was super cool because it was just... they even started off with, like, this is the physiology of these animals. They're able to survive at these temperatures.
Yeah.
We know the fungus doesn't survive at higher temperatures, so we just artificially increased everyone's temperature. The frogs are fine, but the fungus isn't. And actually, like, they have some really nice figures. I have to quickly find which one it was. But there's these nice differences. I think it's this one. Yeah, so the prior exposure ones are in this lighter color, and the naive exposed are in this dark color. And so this is time to clear infection. And then you see, like, there's already, like, infection intensity. It doesn't get as high of an infection level in the ones that have had this prior exposure versus the one that's been first time.
Does this relate... This is infection intensity, though. So what about survival rate?
The survival rate also was beneficial. Here we go. Survival percentage. Here it is. Look at that.
That is one good-looking graph, you guys.
Colorblind friends, this bar here is the control, and that's the prior exposure to heat.
So the heat control just means...
Hello there.
Essentially, that's the untreated group.
Yeah.
That's
they've
group.
gone into the same room with the heat, but they haven't had any of the infection.
Yes. And so this bar here is the percent of animals that are alive at the end of the study. So you want that bar to stay at 100%. That means nobody's died. But then the lower this little step ladder business you see, like the little staircase going down, that means your population is dying off.
And the bottom bar is time in days. So you've taken it out to 100 days.
And there's more than 75% of the population is still alive.
If not in the 90s, I can't quite tell, but it doesn't even...
It's not the 90s, but...
At least high 80s. Something like that. It's a very good result.
It's an impressive graph. As a lady, I appreciate a good graph, and I will say, hats off to them. This is a beautiful freaking graph.
So that's the big takeaway, is that they just have, like, the ones that they did are, like, a little bit more, like, in this figure, a prettier build that they had with the stone. But they had in the... I didn't have a picture of it in the methods, in the supplementary, which I was disappointed of. They were just, like, the raw data. I was hoping they'd have, like, the image of the PVC one that you can build, too, like the cheap one. But there's a description of the methods of how to do the build and what parts you need and how much there you need. And it was just really cool that it's a cheap, fast, affordable solution that not only clears the fungus, but also immune primes the animals.
That is cool.
So for reinfection is less likely. So if you just build these sites, I mean, I guess from the practical perspective, how likely are they gonna, like, to go into the little hut of heat?
Well, I don't know. They might warm themselves, but I don't know. It still seems like an interesting solution.
Yeah.